we shall discuss the biosynthesis of steroids. Now, as in most of the natural product synthesis, biosynthesis, in the case of biosynthesis of steroids also, the starting material is acetate or acetyl-CoA. Now, this acetyl-CoA is a two carbon compound. From that, the system builds up and forms the 30 carbon or 27 carbon or the macromolecule steroid. Now, there are different pathways proposed for the biosynthesis of steroids. We shall discuss here the mevalonate pathway. In a mevalonate pathway, the first step involves the synthesis of mevalonic acid from acetyl-CoA. Mevalonic acid is a 6-carbon compound and acetyl-CoA is a 2-carbon compound. So from the 2-carbon acetyl-CoA, we get mevalonic acid. In the second step, the mevalonic acid is converted to isopentenyl pyrophosphate or IPP. In the third step, the isopentenyl pyrophosphate is converted to Farnesyl Pyrophosphate. Now, Farnesyl Pyrophosphate is a 15-carbon compound. Now, Farnesyl Pyrophosphate is then converted to squalene, a 30-carbon compound. And from squalene, cholesterol is synthesized. And it is from this cholesterol that various other steroids are formed. So, if we can say that cholesterol becomes the starting material or precursor for the synthesis of the rest of the steroids. Okay, so it starts with acetyl-CoA and then we get cholesterol, and from cholesterol most of the other steroids are synthesized. Now, this is the mevalonate pathway. We shall discuss much more in detail what mevalonate pathway and each step is. Now, let us take the first step, that is the synthesis of mevalonic acid. Now, in the synthesis of mevalonic acid, we start with acetyl-CoA. Now, acetyl-CoA, as you can see, is a 2-carbon compound. Along with acetyl-CoA, or you can say acetyl-CoA condenses with melonyl-CoA. Melonyl-CoA or melonyl-CoA. Now, melonyl-CoA is a 3-carbon compound. Now, these two compounds condense together. Of course, everything happens in the presence of enzymes. Alright? Any reaction which is taking place in the living tissue happens in the presence of enzymes. So, here also it is the same case. So, this 2-carbon and 3-carbon compound will condense together to form acetoacetyl-CoA. Okay, acetoacetyl-CoA is a 4-carbon compound. So, 1 carbon atom from this acetyl-CoA, melonyl-CoA condensation is lost as carbon dioxide. You can see here. So, we get acetoacetyl-CoA, which is a 4-carbon compound. Now, this 4-carbon compound again combines with one more acetyl-CoA, one more 2-carbon compound. Okay, in the presence of enzyme again, and forms 3-hydroxy-3-methyl-glutaryl-CoA. Now, this 3-hydroxy-3-methyl-glutaryl-CoA is an enzyme-bound intermediate. You can see here the enzyme E written here. That is, this is an enzyme-bound intermediate. Now, this compound is a C6 compound. It's a 6-carbon compound. Here, because 4-carbon acetyl-CoA and 2-carbon 4-carbon acetoacetyl-CoA and 2-carbon acetyl-CoA combine together and form 6-carbon 3-hydroxy-3-methyl-glutaryl-CoA. Now, this enzyme-bound intermediate then undergoes reduction in the presence of NADPH to form the mevalonic acid. You can see here the mevalonic acid again, which is a 6-carbon compound. So, this is the first step involved in the mevalonate pathway for the biosynthesis of steroids. Now in the second step we have the conversion of mevalonic acid to isopentenyl pyrophosphate. Now we have the mevalonic acid. In the presence of two ATP molecule, it undergoes phosphorylation. Okay, so you get this phosphorylated compound. The OH is phosphorylated. OH of mevalonic acid is actually the primary OH of mevalonic acid is phosphorylated. And then another ATP, in the presence of another ATP molecule, the other OH is also phosphorylated. That is, the tertiary OH is also phosphorylated. Alright, so we have used three ATP molecules. Now, this phosphorylated compound gets converted to isopentenyl pyrophosphate with the loss of one carbon dioxide. One carbon dioxide molecule is lost. So you start with 6-carbon mevalonic acid and you get 5-carbon isopentenyl pyrophosphate, because here one carbon is lost in the form of carbon dioxide. So mevalonic acid undergoes, the OH of mevalonic acid is phosphorylated and one carbon atom is lost in the form of carbon dioxide and we get isopentenyl pyrophosphate. Now in the third step, that is conversion of isopentenyl pyrophosphate to Farnesyl pyrophosphate. Now, isopentenyl pyrophosphate isomerizes in the presence of an enzyme isomerase to form dimethyl allyl pyrophosphate. Okay, now these two isomers, the isopentenyl pyrophosphate and dimethyl allyl pyrophosphate, condenses and forms a 10-carbon compound. Now this is 5-carbon compound, 1, 2, 3, 4, 5. Okay, 1, 2, 3, 4, 5. So these two condense together to form 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10-carbon compound. Now this 10-carbon compound combines with one more molecule of IPP, okay, isopentenyl pyrophosphate, to form Farnesyl pyrophosphate or FPP. Now when you write Farnesyl pyrophosphate in this form, instead of writing it in the straight line or long chain form, when you write like this, you can find here the slight formation of ring A and ring B of cholesterol. Alright, now next step is this Farnesyl pyrophosphate is to be converted to squalene. In FPP, if you consider the pyrophosphate part, moiety, as the head and these two methyl group part as a tail. Now, when FPP is to be converted to squalene, there will be elimination, or in the sense, the FPP will be combining head-to-head condensation. Two FPP molecules will be combining in the head-to-head condensation format. That is, this part, the pyrophosphate moiety, this part undergoes condensation. So this FPP is a 15-carbon compound. So two FPPs combine together to form 30-carbon squalene. So here you can see these head-to-head condensation happens. This particular area undergoes condensation and you get squalene. So you can see here they have condensed from head-to-head in the head-to-head fashion. A squalene is a 30-carbon compound, and here you can see here ring A, ring B, ring C and ring D of cholesterol appearing. Now the pyrophosphate group is reduced with NADPH. The radicals is reduced using NADPH. Alright, now next is the squalene is to be converted to cholesterol. Now, squalene is a 30 carbon compound. It is an open chain compound. If you just see, it is an open. It's not a ring compound. It is an open chain compound. So ring closure has to happen. Now we have the squalene. If we number squalene, in order to understand better how the reaction or the ring closure and other migrations or other reactions are happening, let us number the squalene. We'll start with the numbering this pattern of cholesterol. We'll start with carbon 1 here, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Okay, let's stop over here. Now, before the ring closure happens in the presence of cyclase, enzyme cyclase. Now, before ring closure, the methyl group on carbon 14 over here, you can see here, this methyl group migrates to carbon 13. Alright, the methyl group on carbon 14 migrates to carbon 13, and methyl group on carbon 8 migrates to carbon 14. Alright, I'll repeat once more. Methyl group on carbon 14 migrates to carbon 13, and methyl group on carbon 8 migrates to carbon 14. And also, in the presence of the enzyme hydroxylase, carbon 3 gets hydroxylated. That means an OH group will be attached to carbon 3 over here. So in the presence of cyclase, cyclization happens, and in the presence of hydroxylase, OH is introduced on carbon 3. Before these happen, migration of the methyl group from 14 to 13 and from 8 to 14 happens. Now, for hydroxylation, the hydroxylase enzyme uses molecular oxygen. Alright, now when this happens, you get the compound Lanosterol. Now, Lanosterol has got almost structure similar to our cholesterol, not exactly, but almost the ring, etc. We have got the 3 6-membered ring and 1 5-membered ring has been formed. Now, in the next step, a methyl group is lost. The methyl group on carbon 14 here, the methyl group on carbon 14 gets oxidized and is lost in the form of carbon dioxide. Okay, carbon dioxide is produced. This methyl group is lost and forms carbon dioxide. And you get 14 desmethyl Lanosterol, that is Lanosterol without one methyl group. Methyl on the 14th carbon is lost, 14 desmethyl Lanosterol. Now again, from here, 2 more methyl groups are lost, again in the form of carbon dioxide. From carbon 4, here, these 2 methyl groups are lost. And you get zymosterol. Alright, this is zymosterol. So from 14 desmethyl Lanosterol, 2 more methyl groups are lost and we get zymosterol. Now this zymosterol gets converted to 7,24-cholestadienol. Now that happens by, there will be a migration of the double bond. You can see here we have a double bond over here. Now between C8 and C9, this double bond migrates to C8 and C7 over here. See here, you can see here C8 and C7. So zymosterol gets converted to delta 7,24-cholestadienol by the migration of double bonds from C8, C9 to C8, C7. Now, from this 7,24-cholestadienol then gets converted to desmosterol. Now, in this desmosterol, there is again migration of double bond. Now migration of the double bond migrates from C8, C7, C8, C7. It migrates to C5, C6. It migrates to here. See, goes to carbon C between C5 and C6. So from C8 to C7, it migrates to C5 and to the carbon C5, C6, as in the case of cholesterol. Now from the desmosterol, it undergoes a reduction and forms the cholesterol. Now reduction happens over here, this double bond on the side chain on carbon 17, it's lost. So you can see here the double bond is lost. So it undergoes reduction and we get cholesterol. And from this cholesterol, all other steroids are formed, be it with carbon less than number of carbon atoms less than cholesterol or more than cholesterol. All other steroids are formed from cholesterol. You would have seen formation of testosterone, progesterone, etc. Most of the steroids which we had discussed, all those steroids can be synthesized from cholesterol. So in biological system also, the rest of the steroids is formed from cholesterol. So basically steroids, most of the steroids are formed from cholesterol, and cholesterol is formed from acetyl-CoA. So these are the steps involved in the biosynthesis of cholesterol. Hope it's clear to you. Please do write the steps and make yourself thorough. Thank you so much.